Hi, my name is James George, and I'm an undergraduate in the Biomedical Engineering Department at the University of Michigan. Today, I'll be presenting my Capstone project, which is focused on studying the interactions between host organisms and their microbiota in comorbid conditions of IBD and CDI. I first got involved with this project when I started as a research assistant in the Young Lab in my freshman year of college. For the last 3 years, I've been working on exploring host microbiota relationships, and I've helped to create multiple experiments and models. For my capstone project, I chose to focus on a specific set of experiments that allowed me to study the connections between IBD and CDI. So before we go too much further, I want to provide some of the background of my project and explain a few of the terms that I'll be using. Clostridioides difficile, or as it's widely known, C. diff, is a spore-forming bacteria that causes the highest number of healthcare-associated infections in the developed world. Most healthy people are naturally resistant to C. diff infection, or CDI, since they have an intact intestinal microbiota population. And when I talk about intestinal microbiota, I'm referring to the indigenous bacteria that live within the gut. Studies have shown that an intact intestinal microbiota population provides resistance to C. diff colonization. Now, since antibiotics disrupt the gut microbiota, CDI occurs mainly in patients who are receiving antibiotic treatments. The severity of CDI ranges from mild to life-threatening, and its symptoms can include diarrhea, fever, nausea, and in some cases, death. Part of the response to C. diff is caused by a toxin that the bacteria produces. The image below shows a colon that has been infected with C. diff, which results in a condition called pseudomembranous colitis, which is accompanied by severe inflammation. However, there are factors other than antibiotic treatment that can cause increased susceptibility to C. diff, such as inflammatory bowel disease or IBD. IBD is a condition that affects more than 5 million people around the world, and it manifests itself in two main forms: Crohn's disease and ulcerative colitis. As the name suggests, both diseases result in chronic intestinal inflammation, with symptoms mirroring those caused by C. diff, from abdominal pain to bleeding and diarrhea. Crohn's disease can cause inflammation anywhere along the GI tract, while ulcerative colitis mainly affects the colon and rectum. Studies have shown that people with IBD are at increased risk for CDI, even when they're not being treated with antibiotics, which led to the main question for my experiment. What causes IBD patients to have increased CDI susceptibility outside of antibiotic exposure? So the model shown here demonstrates the three potential cases that can occur. As stated earlier, antibiotic-treated and IBD patients are both susceptible to CDI, while people with an intact intestinal microbiota, which we would refer to as healthy, remain resistant. The lab that I've been working in has developed a mouse model of these three relationships between IBD, antibiotic treatments, and CDI. The overall goal of my project was to use a mouse model created by my lab to ask some questions about the host microbiota responses. In order to do this, we triggered IBD in a group of genetically susceptible mice that are normally colonization resistant to C. diff, and that normally do not develop intestinal inflammation spontaneously. And then using the same type of mice, we treated a second group with antibiotics for 10 days. We then challenged both groups of mice with C. diff spores at the same time point, and then monitored them for colonization and signs of disease. After the experimental data was generated, it was analyzed using R. By using this programming language, we were able to take advantage of different statistical tools and measures to evaluate the data and potential relationships. Reflecting back on the capstone process overall, learning R was actually one of the most rewarding things that I was able to do. As far as biostatistics tools go, it's an excellent one to work with, and it's one of the tools I hope to use in my future career. Now, returning back to the project, the big question that we're trying to answer with these models is what makes IBD-affected patients susceptible to CDI in the absence of antibiotic exposure? In order to answer this question, I constructed two main aims. Aim one involves studying the changes in gut microbiota diversity in the two CDI models. This was done because lower gut microbiota diversity is associated with increased CDI susceptibility. Therefore, I hypothesized that diversity would decrease in both the antibiotic and IBD groups. My second aim was created in order to compare the degree of C. diff colonization and the level of C. diff toxin produced in the two models. From previous experiments with our mouse models, we know that IBD-induced CDI results in decreased disease severity when compared to antibiotic-induced CDI. Using this knowledge, I hypothesized that the C. diff colonization burden and the toxin levels would both be lower in the IBD group than in the antibiotic group. Here's some of the data that was produced from the experiment. And before we look at the trends, I'll explain what the graphs represent. The y-axis for both graphs display the Shannon diversity score, which is a measure of the diversity of a particular ecosystem. And in this case, the higher the Shannon score, the higher the microbial diversity. The x-axis show us time. So overall, these graphs show us how gut microbiota diversity in our mice changed across time. I've outlined the day 0 time point on both graphs, since that's when both groups were challenged with C. diff spores. As you can see, while diversity decreases for both the IBD and the antibiotic groups, the IBD group has a much greater spread or variability at day 0. Now, the next two graphs compare the degree of C. diff colonization and toxin levels in the two models. The colonization burden, which is plotted on the left, was measured by plating the fecal contents at the experimental endpoint and counting the number of colony-forming units, or CFUs. While the difference colonization burden between the two models was not significant, as can be seen by this value right here, we noticed that the IBD mice had a much greater spread or more varied colonization burdens than their antibiotic counterparts. On the toxin chart, which is on the right, we can see that the amount of active toxin is much higher in the antibiotic group than it is for most of the IBD group, and that value is significant. So to summarize, the first question I asked was if the gut microbiota of the two groups were altered. And what I found was that while both groups had decreased diversity, the IBD group had a more variable response. For our second aim, I found that while the differences in the degree of colonization between the groups was not significant, there was a greater amount of variance once again in the colonization burden of the IBD group. Interestingly, we also found that although both groups were colonized to the same extent, the toxin level was much lower in the IBD group. In conclusion, I found that both models were susceptible to C. diff infection, which is what we expected, but that there was a greater amount of biological variability in the IBD model. And this is of particular relevance and importance since it's representative of what's actually observed in human patients in hospitals. These findings help confirm our use of the mouse model for asking more questions about IBD-induced CDI outside of antibiotic exposure. And so from here, our next steps are to dig a little deeper into the microbial populations of each of our treatment groups to discover why they exhibited different phenotypes in response to C. diff exposure. And to do this, we'll use a tool called LEFSA, as well as 16S rRNA sequencing to discover what microbial groups exist within each of our treatment groups. By doing this, we hope to find out if the makeup of the microbial community is a reason for the differing results between our treatment groups, or if inflammation is playing a larger role in the process. Now, before I end my presentation, I'd like to acknowledge the following people for their help. I'd like to thank Dr. Young for allowing me to work as a research assistant in his lab for the last couple of years. And I'd like to thank Dr. Bassis, Dr. Gulab, Dr. Bergen, and Maddie Barron for their expertise and advice on this project. And specifically, I would like to offer special thanks to Dr. Close, who has been my personal and direct research mentor for the last 3 years. Thank you for your time.